In this video, I will be going over the thiamine riboswitch. I will be going over what it is and its experimental history. So our story begins before we get started, actually, thiamine is also known as vitamin B, and these are something that prokaryotes can create themselves, and through a process we call biosynthesis. And obviously, this is a very important area of prokaryotic um, metabolism. And what scientists noticed was that when you add thiamine into cells that have obviously the thiamine mRNA, which is what this Thi-M is. It's a gene encoding for thiamine. Same with Thi-C. They saw changes in the rates of translation and also transcription of not just genes, but specifically thiamine genes. And TPP, by the way, is just a biologically active form of thiamine, also known as thiamine pyrophosphate. And so scientists, what they did is that they identified a sequence that was found in both mRNAs, and they termed it the Thi box. And they kind of hypothesized that probably like TPP binds itself there to regulate transcription or translation or both, right? This was sort of the thought process, but it's just a hypothesis. Just seeing, okay, there's mRNA and you add TPP, and then there were changes in both translation and transcription. So what they did was they did an inline probing experiment. So basically, what this is, look at the figure here. They add a label at one end. In this case, it's the the 3' end, and then they add RNA's cleavage, right? And obviously, RNA they'll cleave at just one spot on average, right? And they add just enough to where it only cleaves on one spot. So maybe it'll cleave here, right? Maybe it'll cleave here, and basically, if we have two different structures and you run this experiment and you run it on a gel at the end, you'll see this pattern. And if we take a look, let's take a look at structure 2 specifically. Of course, we have our intact RNA, right? That should make a lot of sense. That's a very thick band. But then the band right here, right under it, we would imagine that the RNA is cleaved right there, right? Or somewhere along there. So pretty much you just cut off this 5' end and you get a slightly shorter DNA that migrates a bit further, right? In this case, DNA migration is going this way. And the smaller ones go further, right? But notice that you can't get cuts along the double-stranded sides. RNAs just can't really cleave those places, but it can cleave up here when they're single-stranded, right? And so that's why we see a big gap here, which corresponds to this little arm that's double-stranded. And so we see two of them because obviously there's two sides of it, and then the rest would be, the rest of our RNA. And similarly, we see the same thing with our structure 1. We can see that it's pretty symmetrical, right? These two correspond to these two segments that RNAs can't cut at, and the rest are just the other random cuts. And so this is what they did, but with the Thi-M mRNA sequence. And what they did was they ran 5 lanes, and we can see the results here. Lane 1, they pretty much just added the mRNA and TPP, and in the second lane, they didn't add TPP. So this is just seeing, okay, once we add TPP, what changes to the structure? Is there any difference? And I'll go over the results in a bit. Lane 3 was you just added mRNA without any cleavage, right? Kind of acts as like a sort of negative control, right? Making sure that the RNA does in fact exist, and yep, you see the. Oh, I should get my laser pointer. You can see there that it most certainly does exist, so that's great. Lane 4 is we also add OH minus, so hydroxide. And what hydroxide does is it basically allows RNAs to cleave at every single point, regardless of double strand, cleavage, or double strand formation. And does this in a combination of denaturing the mRNA, but also just through like other mechanisms. And you can see that, okay, yeah, it's very, very consistently, just everywhere pretty much. And in lane 5, they used RNase T1, and this just cleaves at every single G, regardless of double-stranded or single-strandedness. So wherever there's a G residue, it'll cut. So that also just gives some better hints. Obviously, we already knew the structure at this point, and so this was just seeing how the structure is different. So lane 1 and 2, if you take a close look here, they at first glance they look pretty similar, but there are a few slight differences, especially in terms of bands. You can see that band thickness varies quite a bit, especially here as well. Down here a little bit as well, right? Over here, a bit of difference over here. Obviously, these could be random, but they are quite a big difference. And so this suggests that, yes, TPP does change the conformation somewhat. Not a lot, but it does change it somewhat. But the most important part is that this SD region, also known as the Shine-Dalgarno sequence, right? This is where ribosomes bind. We can see increased cleavage when there's no TPP. And that is very, very important, because if you imagine, if there's no TPP, then if it's getting cleaved, that means it's single-stranded, right? And if it's single-stranded, that means ribosomes can bind on there to translate, right? However, if there is TPP, and there is double-strandedness at the Shine-Dalgarno sequence, as you can see in this figure, well, ribosome can't really do much there, right? It can't read that sequence, and so this was a hint as to the regulatory mechanism of TPP, and so they continued doing some more experiments, and they made two constructs that they called the translational fusion and the transcriptional fusion. And basically, what they did is, okay, they have the Phe-M promoter, and then they also have the 5' UTR. So this is just like everything before the actual genes, right? If you imagine that maybe there's some promoters there's some operators, there's just some sequences to get, RNA ribosomes, primed, right? Or sorry, not ribosomes, RNA polymerases, primed right before they start translating. That's why it's called the untranslated region. And this obviously includes the ribosome start site. And also includes the Shine-Dalgarno sequence. That's what this RBS stands for. It stands for ribosome site. And so the first one's a translational fusion. And if you take a look at this one, this is basically just our mRNA minus the actual genes. But it's just like the mRNA that should have the regulatory sequence in there. And then right after we have a LAC-Z gene. And remember, this encodes for beta-galactosidase. And this can be assayed for activity. We can either use XGAL, we can also use lactose growth experiments, right? There's a lot of ways to assay for beta-galactosidase activity. And so this basically just couples translation of what would have been, the thiamine enzyme for that Phe-M encodes for, but instead we just replace it with LAC-Z. All right. So if we're gonna get translation normally with the Phe-M mRNA, we're gonna get beta-galactosidase. We can test for that. Similarly with the transcriptional fusion, we have the exact same thing, except we don't put the ribosome and the ribosome stuff on the Phe-M 5' UTR. We instead put it on the LAC-Z gene itself, so that it doesn't change, right? This basically just kind of acts as like making sure that they do get transcribed together. Just to see a bit more of the mechanisms of how this works, right? So if you imagine here, if when you make this transcriptional fusion, no matter what the 5' UTR of Phe-M does, you can still get translation of LAC-Z, and we can assay for that activity. And so consistent with what we've been thinking, right? The translational fusion, in the absence of TPP, right, we got LAC-Z activity, which kind of implies that this ribosome binding site was available to be bound only when there was no TTP bound there. And that's sort of what we saw before, right? We kind of guessed that, okay, when there was no TPP, we got cleavage at the shine diagonal sequence, kind of implying that it was single stranded. And here we actually see that ribosomes can indeed bind when there is no TPP. That's basically what this experiment was showing. And obviously in the transcriptional fusion, you got laxate activity regardless, right? Doesn't matter if you add a TPP or not, which kind of makes sense, right? Because this does not change based on whatever happens on this guy, right? So that's all good. And before we move on, let's just recap what we know so far. We know that in the presence of TPP, we get double-strandedness at the shine diagonal sequence, preventing ribosome binding and therefore preventing translation. Contrary to that, when there is no TPP, right, no thiamine, we see that there's single-stranded shine diagonal sequences which lead to translation, right, because the ribosome can bind, and now it's confirmed in the experiment where we made two constructs, and the translational fusion, we were able to see that indeed there was translation because when there was no TPP, this ribosome binding site was open, and that ended up translating the laxate gene, therefore producing beta-galactosidase, which we assayed for. Okay, so that's all great, but we don't actually know what the conformational change of this ribosome is once we add or we don't add TPP. And so that's sort of what we, with the not me, with the scientists were trying to look for later on, and they did this by mutating base pairs, right? So this was just to see how this double binding was behaving, and okay, the figure on the right is very, very much just ignore most of the mutations. The ones I want you to look at are M1, M2, and M3, M4, right? Just these two regions over here. When they induced M1 and M3, right? So M1 would be over here, changing both bases at the bottom to a U, right? You see they changed these two A's into a U and a U. And likewise for M3, what you can see is that they changed these two sequences, the G and the U, into a C and an A. And basically that would just prevent double-strandedness, right? You can see that over here, there is a G bound to a C, which is great, and a U bound to an A, which is also great. But we basically, with M3, we blow up that binding, right? And we see that once you do that, once you make those mutations and you break these double strand bonds, you actually don't get TPP regulation anymore. Doesn't matter if you add TPP or if you don't add, there is no regulatory effect. So that was very key in knowing that, okay, it is in fact the conformation that is causing this transcriptional or translational regulation. Most likely translational, right? Because we saw earlier that transcription still worked perfectly fine. So that's a very good hint. But then also very important is that when they added in the M2 and M4 mutations, right? If you take a look at M2 and M4, you can see that they basically just swapped the base pairs, right? They instead of the GU on the left, they put it to the right, right? And instead of the CA on the right, they put it to the left. And so this maintains the double-strandedness of these RNAs. And they saw that, yeah, this rescued M1 and M3 mutations. That this worked just as well as normal. And so this suggests that it doesn't really matter what bases are actually pairing, so long as you see pairing anyway, right? I guess they didn't really test for like differences in pyrimidines or purines, right? But at the end of the day, the take-home message is that it doesn't matter if it's C and a G or A and a U, so long as there is double-stranded formation. That's all you need. Now, this is where they really started to see something very interesting. If you take a look at this M8 mutation up here, where they changed the U into a C, right? If you imagine what happens when you change the U to a C, you would end up getting better double-strandedness here, right? Because a C would bind to a G. And so this should stabilize this area up here. And that's exactly what we see. We see translational inhibition. And this is exactly what the shine diagonal sequence is, right? And so if we're strengthening this double-strandedness at the ribosome binding site, Yeah, we should get translational inhibition. That kind of makes sense, right? The ribosome wouldn't be able to bind here because we strengthened this thing, this double-stranded interaction with using M8. And so this is where we actually get our model, because we're just going to investigate this mutation a bit more in depth, because we can see that once you make this mutation, we get both stabilization of this conformation, right? This is kind of what we were saying before, right? We have our shine diagonal sequence that's a bit more stable than before, but we also get destabilization of the other conformation, where the U would normally be bound to this A, right? And so this sort of suggests that there is a change in conformation where the strand that would usually bind to the shine diagonal sequence actually binds to a different place on the mRNA, opening the shine diagonal sequence. That was the sort of the key breakthrough. And inline probing also showed that when you add this mutation, you reduce cleavage at the shine diagonal sequence, which kind of just like more confirms that, yeah, it does form double-strandedness at the shine diagonal sequence. And so then we get to our final model, right? This is the same image as before, except I removed the mutation. Is that when we add thiamine, or TPP, we get this shift in conformation of our mRNA. Remember, this is the mRNA 5' UTR, right? This isn't really translated at all, right? You can see our AUG here, and we can see that this is where the upstream genes are. So all of this is all the UTR, the 5' UTR, right? You can even see the 5' end right here, right? And this is what we call a riboswitch, because when you bind to something, you bind to, in this case, TPP, there's a conformational change that now opens up the shine diagonal sequence. And this now allows ribosome binding, so that the ribosome can get like hooked onto the AUG and then start transcribing upstream genes. And so this was what we found, or what the scientists found, and was very groundbreaking, because they started to see that these riboswitches were everywhere in prokaryotic life, even in eukaryotic life, like yeast. They still have riboswitches, right? So this is this was a very key find in translational regulation. And there's also transcriptional regulation, because this can also form like a hairpin loop during RNA polymerase transcription, which ends up attenuating transcription if within the presence of TPP. And this kind of makes sense, because if you already have a lot of TPP, you don't need to make more TPP, right? So TPP will bind to this mRNA to kind of tell the mRNA, hey bro, we have enough TPP. You don't need to make more. Blocks off this shine diagonal sequence and prevents the waste of resources to produce more of these genes involved in TPP biosynthesis.